Okay, I'm going to go through the answers to the questions that I set on the nervous system. So we start with question 2. Animals receive different stimuli from their environment. Their synapses can manage multiple stimuli, often resulting in one response, such as muscle twitching. So here's the clue to this question. You've got multiple stimuli resulting in one response. So what we need to do there is therefore look at a diagram where we have neurons, multiple neurons, okay, with messages travelling in from different areas resulting in one response. And so this, we know, is called spatial summation. So the answer is A. Okay, so let's go on to question 3. So it says, which of the following describes the process that happens during repolarization of a neuron during the action potential? So just a quick reminder of an action potential, and repolarization is this section here, okay, where we're becoming more, or relatively, it becomes relatively more negative, okay. So if we remind ourselves of what's happening in the axon, okay, at that point there, we've got potassium ions on the inside of the axon, and actually for us to become relatively more negative, those potassium ions have to leave through those gated channels. And so we're going to have our potassium ion gated channels are going to open. And so let's look at our answers. Now this one's nice and easy to start with. The membrane potential we know is decreasing, so it's definitely not C or D, it's going to be A or B. And we know that our potassium ions need to flood out, and so our potassium channels or ion channels are going to be open, and so the answer is going to be A. Right, question 4. The figure represents the voltage across the membrane of an axon and the changes that take place during the generation and transmission of an action potential. So let's have a look at our picture. Okay, so they've just given us what we've just drawn. So indicate the letters A to G where each of the following occurs. A letter may be used once, more than once, or not at all. Okay, so voltage gated sodium ion channels are open. So that's when those sodium ions start flooding into the membrane. So that's going to be happening here at the start when we've had a stimulus, and here. Okay, so the answer is going to be B and C. Repolarization, here, we've just looked at that already. E. So it's going to be this very top bit here, because those sodium ions are going to start flooding in until we reach this bit, and then we're going to have a fall. Okay, so the answer is going to be D and E. And then sodium ions are actively pumped out of the neuron. Well, we know that that's used to establish the resting potential. So we've got our resting potential here, A and G. So it's definitely A and G, but also in F, because we're just getting to the point where we are re-establishing that. So A, F and G. Okay, question 5. Synapses are an integral part of the nervous system. Outline the roles of synapses in the nervous system. Okay, so when we know that they are a junction between neurons, so they allow neurons to communicate. We know also when we talked about them that the synapse can only pass information in one direction, so they allow for messages to be unidirectional. We also know at this point here that when we get to the synapse, we know that if we don't have enough of the neurotransmitter, then the impulse in the next neuron isn't triggered. So it allows there to be a threshold so that any kind of low-level stimulus isn't going to trigger an impulse. It's going to kind of get blocked out. So the threshold allows low-level stimuli to be blocked. The other things that we can add there actually are when we talked about summation and when we talked about multiple neurons meeting on one neuron, it allows for us to have convergence where we have more than one neuron meeting to give one response. So you can say that it allows convergence, but it also allows the opposite of that, where we have one neuron which we know then sends various responses off in different directions. So it also allows for divergence. Okay, so let's move on to question 6. So figure 3.3 is a diagram representing the neuromuscular junction in mammals. So because it's neuromuscular, what I've done is I've labelled this half we know is the muscle, and here is the neuron. And they've given us various symbols, so let's have a little look at what these are. So here's our axon, our myelin sheath, we've got mitochondria, we've got our vesicles containing acetylcholine, then we've got W, we've got our synaptic cleft here in between, then we've got X and we've got Y. So what type of molecule forms ion channels W and X? So any kind of channel, we're looking at channels through the plasma membrane, the bilipid layer, so we know they're going to be made of protein. Identify region Y. Okay, so this is the synapse, but it's this, the whole thing, sorry, is the synapse. This is referred to as the synaptic cleft. And you have to have cleft. You can't put gap, they just won't accept that answer. Y, name the enzyme found in region Y. Well, we know, they've told us that the neurotransmitter up here is acetylcholine, and so we know that the enzyme that breaks that neurotransmitter down once diffusion has occurred and the muscle has been stimulated is going to be acetylcholine esterase. Okay, question 7. Outline the ways in which the structures of a sensory neuron and a motor neuron are similar. Okay, so let's have a look at those. I'm just going to draw a quick little diagram just to remind me. So sensory, remember that the cell body is always in the centre. That's how I remember that. Okay, these are not very good diagrams, but they will do. You could see that if I win my A-level exam, this is exactly what I would do. Just take me a couple of minutes, just so, well, not even a couple of minutes. There we go. Here's my motor and here's my sensory. Okay, so now we have to look at the similarities between the structures of these two neurons. So now this is almost like spot the difference, but spot the things that are the same. So they both have a cell body. Both have an axon. There it is. They both have myelin sheath. Or I could put there Schwann cells, because we know that the myelin sheath is formed by Schwann cells. I could also say that they both have dendrites. At the ends of the neuron. Okay, nice and easy, 4 marks there. Okay, question 8. Squid are an order of aquatic animals. Many species of squid show a reflex position to changes in their body position. A sensory receptor called a statocyst detects changes in the body position of the squid in the aquatic environment. The reflex response corrects the squid's body position. Describe the components and events involved in a reflex arc. So there's no different to GCSE. You've got to think school's really mad. Okay, so as in the order of the neurons. So we have a sensory neuron, which feeds into a relay neuron, which is part of the central nervous system, so usually the spinal cord, and then we have a motor neurone. Okay, and so we know that in between those we would have a synapse. Synapse here, synapse there, and at the start we would have here would be our statocyst, which is where our stimulus is first detected. Okay, so I mean I've just written this in shorthand here, but you would just need to write this in order in a couple of sentences to pick up those marks. Okay, part 2 suggests one benefit to the squid of the reflex response described above. Let's remind ourselves of that reflex response. So the reflex response corrects the squid's body position. Okay, let's look at this again. A sensory receptor called a statocyst detects change in the body position of the squid in their environment, and the reflex response corrects the squid's body position. So if you think about a squid in the water, why might they need to change their body position? Well, it might be for balance, or it just might be as they're moving around, it provides much better or more efficient movement. Okay, part 3. A statocyst sensory receptor is shown in figure 2.1. What can you conclude from figure 2.1 about the mechanism by which a statocyst acts as a transducer? Let's just remind ourselves a transducer is where energy, or how energy is transferred, or where it's transferred. Okay, so we're just looking at energy transfer. So let's go back to this and make sure we understand it. So here's our neuron. We've got an epithelial cells all the way around, and then we have little sensory hairs, and we have this statolith, which is a mineral, so some kind of calcium, I would guess, mass that can move within the statocyst. So this is a ball basically that's just going to move around within this gap here, and as it moves you can see what it's going to do is it's going to push on these sensory hairs which attach to these epithelial cells, and then that is going to trigger the impulse. So what can you conclude about the mechanism by which the statocyst acts as a transducer? Okay, so what's happening in terms of energy here? The first thing here is that the statolith is going to move, and it's going to move the hairs. So any kind of movement, that's going to be kinetic energy. And then that's going to trigger the action potential, which we know is electrical energy. Okay, so let's move on to the next question. So it says, lobsters and sea cucumbers also have statocysts. From this information, the student concluded, I think this means squids, lobsters and sea cucumbers have evolved from a recent common ancestor and are probably classified in the same phylum. So, the question says, evaluate the extent to which the information about lobsters and sea cucumbers supports the student's conclusion. So when we see the word evaluate, in our answer we have to give a judgment, and then we have to give reasons for and reasons against. So I'm going to say that my judgment is that it doesn't support the conclusion, and I'm going to give my reasons against. Well, there's two. The first one is that if we look at the phylogeny, okay, which is how things are classified, clues in the gen bit there, we're looking at the genetics, and really we can't make a statement that they had common ancestors until we have looked at the genetics in more detail. So phylogeny means we need to examine the genetics. And the second reason is that actually this could be an example of convergent evolution. Now, convergent evolution means that is when features appear in different species without there being any common ancestors. So if you think about wings, for instance, bats have wings and birds have wings, but their wings didn't evolve from the same ancestor. They actually just spontaneously evolved in those different species. And so this could be an example of convergent evolution. Reason 4 is that they do have similar looking features. But for me, that wouldn't be enough. And I would say, my judgment says I don't support this conclusion because of these 2 reasons. Okay, question 9. Multiple sclerosis is an autoimmune disease that damages the nervous system. Suggest how the autoimmune system causes damage to the nervous system. So we have to include nervous system. We have to relate our immune knowledge or immune system knowledge to the nervous system in this question. Now, an autoimmune disease, okay, is when the immune system is unable to recognize self. Okay, so it's not able to recognize its own cells and sees these as foreign. So when that happens, it kicks in with the cell-mediated response, which involves the T lymphocytes. The cell-mediated response is when own body cells which have mutated or been infected with the virus are attacked to be destroyed. Because this is still part of the body, we still kick in with the cell-mediated response, even though the body doesn't recognise these as our own cells. So the T cells would attack the neurons, and that's my bit that I'm relating it now to the nervous system, and they would also release cytokines. Cytokines are basically chemicals that are used to signal between different cells. They're a bit like hormones, but because the amounts that are released are so small, we don't actually refer to them as hormones, we just call them cytokines. Now, there are particular cytokines that are released in the immune system, and they are called interleukins. Now, if you think about that, inter means between. Leukins, we're referring there to leukocytes, which are white blood cells. So we're looking at chemicals being released between white blood cells. We've got some signalling going on there. And what that does is it stimulates B cells to divide, and they then produce antibodies, and those antibodies attack the neuron. Okay, next question. So, figure 5 below and on the insert shows 3 neurons of different sizes from a person with MS. So let's have a look at those. So here's our figure. So this one here is our neuron affected by multiple sclerosis. And we've got a little key here. The purple regions, those are the axons. The light blue are the Schwann cells. A little bit there. And then the black is the myelin sheath. So the question says, one of the neurons has been affected by MS. MS causes or changes to neurons which reduce the speed at which nerve impulses are conducted. Using information from figure 5, what can you conclude about how MS causes a reduction in the speed of nerve impulses? So let's just write down first what we know about the speed of impulses. So ordinarily in mammals, we have our axons. Here's our axon, and we have our nodes of Ranvier there, and we have our myelin sheath there. And we know that the impulse, we get impulse jumps, we get this local circuit that sets up between those nodes of Ranvier, and so it jumps fairly quickly down the axon. Okay, so we only get depolarization at those points there. Now we know if we don't have our myelin sheath, then we have to have depolarization at every single point along that axon. So we get loads and loads of little tiny localized circuits setting up, and each one of those means, because we've got so many, it takes much longer. Okay, so this is going to be quick, this is going to be slow. So now let's look back at our picture and the information they provide of the MS neuron and the normal neurons. Well, what can we see? First of all, the points that we're picking out, and then how does that mean it's going to be slower? Okay, so first of all, the point is that there are fewer Schwann cells. There is no myelin sheath that I can see. So now let's do our so. This means that there is no saltatory conduction, which is here. Remember when it jumps from the nodes of Ranviers to nodes of Ranviers. So, and we have very small, short or local circuits required with depolarization along the whole axon. So of course it's going to be slower. Right, question 10. Which of the following diagrams A to D shows a sensory neuron? Okay, so nice and easy. We talked about this earlier. The sensory neuron who's got the cell body in the centre. Okay, there. So that is going to be D. The next question, the table below shows the membrane potentials of different neurons at cholinergic synapse. So that means that we've got acetylcholine there as the neurotransmitter. The data were recorded on 5 separate occasions as shown in the 5 rows. So this was the first try, second, third, fourth, fifth try. So we've got 3 presynaptic neurons, A, B and C, and we've got 1 postsynaptic neuron. So let's just remind ourselves what that's going to look like. So let's say that's our postsynaptic neuron, and then here are our presynaptic neurons. So that's A, B, C. So let's see what's going on. So if B and C are depolarized, we get an impulse in our postsynaptic. So if this one goes, this one and this one, we get an impulse. If A and C do, we get an impulse. If A and B do, we get an impulse. If C on its own goes, we get an impulse. Whereas if we don't have any depolarization in any of them, then we don't get an impulse. So this is going to be an example, therefore, of spatial summation. Okay, because we've got 3 neurons leading into the response in one, and if we get 1 or more of those original neurons stimulated, we will get that response. Okay, last question. Mollusks such as H. lunulata have unmyelinated neurons. Saltatory conduction cannot occur in these neurons. Why is transmission of action potentials along the axon slower in the absence of saltatory conduction? Just as we've done in the last question, it means that we have to have depolarization along the whole axon. It means that we have very short local circuits.